Welcome to the IMBIS Ask Me Anything series. Today we have Dr. Janet Dubinsky joining us. The AMA series is presented by the International Mind, Brain, and Education Society with the aim of building connections among IMBIS trainees and non-trainee practitioners and researchers to support the translation of research into policy and practice. It's also an effort to expand our community and welcome new names and faces into this space. So if this is your first IMBIS event, we are glad you are here. My name is Samira Massey. I'm the trainee board secretary. Just to give you a little bit more information about IMBIS, we are holding our biennial conference this year in Leuven, Belgium, from July 10th to July 12th. So there is still time to register if that's something that you might be interested in. Lots of great things happening there in research and practice. We are also the home of the Mind, Brain, and Education Journal, which provides a forum for the scholarly communication of basic and applied research on learning and development, including analysis from biology, neuroscience, cognitive science, and education. So here's what to expect from the AMA sessions. These sessions are recorded so that they can be shared on YouTube later. Please watch out for an announcement about that on Twitter and our website. If you have registered, you will also receive an email with a link to the recording once it's available. Today, we have 2 ASL interpreters and live transcription is enabled, so please take advantage of those services if they're helpful to you. If you require signing live today, you can also enable the ASL channel. I'm not sure if we have that working, so what we've done is we do have them spotlighted for you, so they should stay on your screen. If you need to change modes with your gallery, then that might help you as well, but they should be pinned there for you. Please let us know if you have any issues with that in the chat, and we'll work with you to make it accessible. We're going to start this session with a 15-minute presentation from our speaker, Dr. Dubinsky, and when I say 15 minutes, we're going to be flexible with that because I know there's a lot of information. So we'll let Dr. Dubinsky talk for a little bit and teach us some great things, and then we will have a discussion and Q&A session from you because it's important to have this interaction. So please make sure that you write down your questions, put them in the chat, and we'll make sure that we get to them. Before we start, there's some quick vocabulary you might need to know. The term MBE comes up a lot. It stands for Mind, Brain, and Education. We have a diverse range of participants. We want for this to be an inclusive environment, so please ask if you're unsure about what anything means. Our team is going to keep an eye on the chat. We'll make sure that we address questions that you have. While I introduce today's speaker, we do have a poll question for you that Chris, our co-president, is going to push out to you. So if you could just take a second to respond to that poll question. But it is really genuinely my pleasure to introduce today's speaker, Dr. Janet Dubinsky. She's a well-known neuroscientist and community educator. She's a professor of neuroscience at the Neuroscience Department of the University of Minnesota Medical School. Dr. Dubinsky received her bachelor's degree from MIT and her PhD in neurobiology at the University of North Carolina at Chapel Hill. She's also a leader in the graduate program in neuroscience. She teaches undergraduate and graduate neuroscience and is the founder of BrainU, which is a professional development program. In 2009, she was awarded the Science Educator Award from the Society for Neuroscience, and she served on the Society's Public Education and Communications Committee. Dr. Dubinsky has led and is currently a member of the graduate program Neuroscience Public Engagement Committee, which runs Brain Awareness Week and Minnesota Brain Bee. BrainU, initially developed with the Science Museum of Minnesota, provides professional development in neuroscience for K-12 teachers using curriculum written for their classrooms. Dr. Dubinsky has documented BrainU's beneficial effects for teachers, classrooms, and students both domestically and in Liberia. The effort has been supported by the National Institute of Health Science Education Partnership Awards, the Howard Hughes Medical Institute, and the Carter Center. She's a major contributor to knowledge generation in the metabolism and physiology of the brain, with particular attention to metabolic abnormalities present in Huntington's disease. She's also internationally known for her research on the impact of neuroscience knowledge on teaching. At UMN, her work and teaching have been recognized with the Outstanding Contributions to Post-Baccalaureate, Graduate, and Professional Education Award in 2011, and the CBS Stanley Dagley Samuel Kirkwood Undergraduate Education Award in 2020-2021. I'm going to pass it over to Dr. Dubinsky. Welcome, and thank you so much for being here with us today.
Thank you, Samira and Kristen, and the entire IMBIS trainee board. Let's see if I can get to sharing my screen and getting you so you can see my slide set. I can do this. Hide the floating controls. All right. And I think it's time to end the poll, if we could, please. I want to talk to you today about my distillation of the cognitive neuroscience literature with respect to active learning and direct instruction. All right, so we've seen the poll. I'm going to turn it off. Active learning is when students participate in some way, shape, or form with the materials. Why isn't this going away? Through either problem sets, group work, discussions, things that you know how to do. But direct instruction is the very traditional teacher as sage on the stage, telling students what the information is, that it's important to learn, and hopefully the students take notes, go home and study it, and then are able to spit it back on the test. Oops, all right, let's see if I can do this. I don't understand why this doesn't go away. So if you could help me, Kristen, to keep those things from popping up, I would greatly appreciate it.
Is it the poll that you're seeing?
I'm seeing things on top of my slide set that would obscure the view for other people. So if that's not obscuring other people's views,
no, we can't see it. We can't see it. So sorry about that.
Then I will continue. So I want to start with a study done in physics classes at the university level, where students were either taught through direct instruction, that is lecture, or they were taught in class through active learning exercises, including group problem solving, individual problem solving, lots of discussion and question and answering. And this was done probably at Harvard, because that's where the authors are from, for two different topics. We're seeing the data for statics, but they also did it for a fluidics unit, and the students had passive learning for one topic and active learning for the other. So the data represent the same sets of students in comparable circumstances, and it also had very good alignment of content between the instructors of the same unit tests. So very clearly designed crossover experiment. And on the end of unit test of learning, the students in the active learning condition performed about 10 points higher on the final test than students in the lectured classes. However, when students answered a poll about where they thought they learned better, not surprisingly, they favored the passive classes, the direct instruction, where they were provided all of the information, and they felt like they learned better under those conditions. So I asked myself, could neuroscience explain in some way, shape, or form this discrepancy in learning? Okay, hold on, we're trying to move forward here. And now I can't move my slides forward. There, all right. So can neuroscience explain this? And the mechanisms that I have come to conclude are the principal ones that we need to be aware of are synaptic plasticity, which is the basis for all learning and memory and everything that goes on in the nervous system, and a large circuit or system within the brain called the reward system. And I can't do a talk without showing you real synapses. Here, there's an axon in red that is called a climbing fiber because it climbs up the cell body and dendrites of Purkinje neurons in the cerebellum, which are expressing green fluorescent protein. And every place you see a red bump, that is the site of a synapse. The red presynaptic nerve terminal is secreting a neurotransmitter, which is then sensed by protein receptors on the postsynaptic dendritic spines or specializations to cause information transfer. The information flowing through the axon electrically is very reliable, but this chemical transmission across the synapse is quite variable and depends on how much neurotransmitter there is, what kind of receptors are waiting on the postsynaptic side, what state the intracellular domain is in, and there's quite a bit of variability there and the ability to change. So it's at these plastic sites where we can manipulate synaptic plasticity and increase or decrease the strength of connectivity by changes in the chemicals surrounding the synapse or the intracellular environment inside the postsynaptic cell. But these are the physical site of all the plasticity that happens in the nervous system. And here's an example experiment of how such learning changes synapses and the circuits of neurons that interact because of those synapses. In this experiment, the mouse was genetically engineered to express a protein that changes its fluorescence when the cell body is active. And the skull of the mouse has been thinned so that a microscope can actually visualize the fluctuations in intensity in those neurons that are visible on the surface. And the mouse has been trained to crawl into this tube on the microscope and lick water from a port when a light cue goes on. And today we're going to, or in the experiment, we're going to see that the mouse learns how to also press the lever in order to receive that drop of water. So on the first training session, the first day, lots of trials in gray, the average in black. The mouse might accidentally press the lever once and probably was quite disappointed because it didn't get a lot of water. And over the course of training, the average amount of lever pressing increases until after 2 weeks of crawling into this device and learning how to get water, the mouse knows exactly what to do and presses the lever every time. So as it's learning this physical movement of the lever, what happens to train the neurons? And each different colored dot on the right here represents the firing of a different neuron in the field of view. So in the first few sessions, when the cue light goes on, some of the neurons respond, but not very many. By the end of 2 weeks of training, when the cue light goes on, lots of neurons respond, and in a very stereotyped, repeatable fashion, so that some respond immediately, others respond with a specific delay, indicating that different neurons are being activated in a circuit that is coordinating the activity of the lever pressing. So this is a real example of building the network activity from the synaptic plasticity. And similar changes in the patterns of neuronal firing occur in some part of the brain whenever a new task or mental activity is learned. So that's what we want to promote in all of our students, is that kind of synaptic plasticity. Well, let's move on then to this reward system or reinforcement learning system. The reward system involves several parts of cortex, not all of which are shown here, but at the heart is this structure called the basal ganglia or the striatum, which calculates how valuable something is. And I don't care whether that something is a physical movement or that something is something to learn or to buy or to pay attention to, or anything you do, whatever is going on, you have to decide, or your brain has to assign a value to it in order for you to make the decision to buy one brand of tomato sauce over another brand of tomato sauce. And so there's inputs from lots of places throughout the nervous system to this very deep and central part of the brain to make these value calculations. Here's another diagram of the same thing where we've cut away a quadrant of the brain, and you can see the basal ganglia deep inside the cortex. And we've diagrammed the long loop pathways which run from cortex to the basal ganglia, and then out of the basal ganglia to the thalamus and back to cortex in a manner that we can track different cognitive and motor behaviors. Within the basal ganglia, there also are loopy pathways from a midbrain dopamine center that go to the basal ganglia striatum and come back to the midbrain. And there, not only do they start and stop in the same place, but they also feed forward to the next area. So you get a spiraling of information across the cortex from medial to lateral as an activity is being unfolded or evolving. Notice also, it's these dopamine projections from the midbrain that are making the value calculations in the striatum. So notice also that there's loops to the hippocampus, which we consider to be an early stage of learning. And so as the values are calculated in the basal ganglia, those values get added to the event or episode that is being stored in memory, and the storage, which goes from hippocampus to cortex, then gets stored with not only the event itself, but the value of the event. So it's a complicated set of interacting loopy circuits, and the basal ganglia, this dopamine, decides basically whether to let information move through the circuit or not. So it acts as a gate to let information pass or not pass, depending upon the value that has been calculated in the ventral striatum or the basal ganglia. So the rewards circuit calculates this expected value of an action, a decision, a sensation, something you remember, some social interaction, but most important for our classrooms, calculating the value of the information being exchanged. And these calculations are based on dopamine interactions, but they qualitatively represent the cost of effort. How much work will it take to do this task? Or how much reward or recompense will I get at the end of all of this effort? How close am I to finishing? What really is waiting for me at the end? And these anticipatory value signals get higher as we get closer, but we get to the end and then we get some kind of outcome and the feedback from that outcome that tells us whether we hit the mark or whether we still have a ways to go to get to our goal. And so there's a discrepancy that's calculated, and that discrepancy informs us that we're not quite there yet, and we have to repeat this. And sometimes we call those discrepancies errors. But the more we repeat this process, the smaller the errors get and the closer we get to our goal. So how does giving students choice or agency in the classroom, as one would in an active learning situation, interact with this reward system? Well, it turns out there's another signal that goes across the striatum that we could measure it in rodents. We don't have the techniques to measure this in humans yet, but in rodents, we can see a signal at the time or after the time of reward move from across these value calculating areas, telling synapses that what happened was valuable enough to remember now, and feeding the transfer of that value signal to the hippocampus. It's important to know that I did it and I got it correct, and for the synapses that got me there to be able to become stronger so that I can do the same thing again the next time. So let's quickly look at two experiments that demonstrate both behaviorally and neurally that that kind of choice or agency in doing an action actually augments learning. Here, human subjects are in the magnet, and they're tasked with learning Japanese characters and what those characters represent in terms of a symbol object pair. And they either are told they get the choice, they can choose which character to study and uncover the matching object, or the computer says, I'm going to tell you which one to study this time. And the participant has to press the indicated lever to look at this particular symbol. When tested immediately or 24 hours later, subjects remember the symbol object pairs better when they had the ability to choose which one to study than when the computer chose for them. The difference being significant, but also similar in size to what we saw in the physics experiment. And moreover, if we look at the signals in the brain, what parts of the brain are more active than baseline when the computer says you get to choose, this ventral striatum basal ganglia area has an increased signal that anticipates a increased signal in the hippocampus at the time the object is revealed and the encoding of the symbol object pair occurs. And that is greater when the subject subsequently remembers the object than when the subject doesn't. And it's greater than when the subject had choices versus when the computer said, study this. The signal in the ventral striatum also predicts how well symbol object pairs will be remembered. So here we definitely have hippocampus and reward system, key structures in the reinforcement learning circuit, being activated to a greater extent when the subject has the agency to actually choose what to learn. All right, here's the second experiment. In this experiment, there's no rewards. It's all self-motivated learning. The human subjects in the magnet are here, two different sentences for which there is a brand new word that they are learning in the context of those sentences. And in some sentences, the new word is used congruently in both sentences, so you can figure out what it means. And in this first set of sentences, the word Jedden obviously means cemetery. But in the second set of sentences, the word Hutel, the meaning is incongruent because what the astronomer is watching may or may not be what he drank in the morning or she drank in the morning. And then again, subjects are given a test afterwards to see what they remember. So we look for activity above baseline, and what becomes prominent is, again, areas in the reinforcement learning system. During the first sentence, the ventral striatal, this key value calculating area, is active at baseline or a little below, but not significantly so. When the second sentence is presented and the word meaning becomes obvious and is subsequently remembered, the information or the content of the striatum and its activation goes up, more for remembered words than forgotten words, and more so than when the words are incongruent and you can't figure out what they are. And that's true for both sides of the striatum. It's also true for this dopaminergic area in the midbrain, and the graphs look identical for the hippocampus, this critical structure in learning and memory. So this is an internal act of extracting value from what went on cognitively in the brain, activating the reinforcement learning circuit through the repetition and the repeated use of the word. So the reinforcement learning circuit is activated in a wide range of contexts where one has agency, when one is curious, when one makes and corrects errors, and when one is engaged in social interactions. I value what is going on in this conversation, and I look forward to the questions you're going to ask. The reinforcement learning circuit makes these value calculations, which are synapses that get stronger depending upon the context, to make learning more salient. And multiple parts of the brain feed through this system so that we can place value on lots of different kinds of activities. So in summary, I want you to take home two big neuroscience ideas to guide your pedagogical choices. Maximize student opportunity for synaptic plasticity. It's their brains that have to change, not yours as the teacher. And for the students to have opportunities to exert agency and create for themselves value for the content you want them to learn. And before we go to the questions, I just want to say one mea culpa, and that is, no experiment that I reviewed in the literature or have shared with you today directly measures the neural activity and synaptic plasticity in the reward circuit activation in students in real world classrooms. We're not there yet. The experiments that I've discussed are all situations that have something going on analogous to what would happen in real world teaching and learning. So thank you very much for your time. I look forward to your questions, and if we don't get to them, there's my email address so you can contact me separately. So thank you very much.
Okay, Dr. Dubinsky, thank you so much. That was extremely, I think it's generated a lot of thought and a lot of questions. And I know it's a lot to cover, and we ask 15 minutes. It's like, hi, cover everything. You have 15 minutes. So it's a tall order, and I appreciate what you've done. So to start us off, I just have some general questions first, and then we've gotten a few questions in through the chat. What do you think is the most impactful change that you've seen from the work that you've done?
Well, the work I've done is teaching neuroscience to teachers. And what I've seen the teachers use is, for many years, my message was synaptic plasticity. And they've taken the various aspects of that synaptic plasticity to justify connecting new knowledge to prior knowledge, giving students opportunities to play with the material in multiple ways over multiple days, to spiral the activity or the content, to space and interleave it, all of which are known pedagogical practices based on behaviors, but the synaptic plasticity increases every time one goes through the material again and connects new material to old and remembers that old and reactivates those synapses. So the teachers told me how they use that information. And in digging into the reward circuit, I hope that that information will further inform teachers' choices of pedagogies.
And I think that really kind of segues into a question that we got through the chat from Ann Fency, where she was asking, what do you mean by increasing students' synaptic plasticity in the classroom? And can you give some concrete examples of what this looks like? And I think you just did that, but I'm going to leave that open for you in case there's anything to clarify or add.
Well, students learn when they're just listening. Okay, we're humans. We do learn from listening, but it's much harder because it requires us to focus our working memory on what's being said and to process it. To follow my description, you had to think hard and follow my logic and my presentation. Maybe you were also trying to take notes. That distracts from your listening. So cognitively, those are two very difficult things that you're doing simultaneously, and it will diminish your ability to remember what I'm saying versus what you're writing down. You have to make some choice one way or another. if you've got students solving problems first individually and then in groups where they share their solutions and they talk about which ones might be better or what justifies this solution over that solution, then they're manipulating the material multiple times in a social situation. So they are going over that material multiple times, hopefully activating the synapses, correcting their own errors as they weigh the value of which way to solve these particular problems. So you're engaging their reinforcement learning circuit for multiple passes through the material. We don't sit students down in the classroom and make them run the maze 1,000 times like we do our experimental rodents. That just doesn't happen. But we have to give the students more than one opportunity to encounter the material and to discuss what it means. And I don't care if you're talking about history or science or character development in a novel. We have to discuss these things. And I might place, greater emphasis on character development, whereas someone else might place greater emphasis on the narrative path the story takes. We analyze those things and we rework the story and we reinterpret it based on the points of view we bring to it. So all of that is manipulation, is the same as activating the value centers in our reinforcement learning pathway. I can't look at a student and say, oh, your synapses are getting stronger today, but not yesterday. Hopefully that's happening every second of our lives. And we can't measure today I'm doing better than I did yesterday. We just have to provide the opportunities and let those processes go and mechanisms go on in the brain, because we're not going to open up the lid and look in. We're just going to have the practices in our classroom that potentiate learning as much as possible. So all of those pedagogies that are considered best practices by the educational psychologists, I think, can be explained by either synaptic plasticity or activity in this reward circuit and the communication through multiple places in the brain. And we don't say, okay, turn your hippocampus on now. Oh, go out to recess and turn your hippocampus off. It just happens. We create the opportunities and the students' brains manipulate the material.
Pauline Chapa had a question, and she was asking if you think cognitive neuroscience with cognitive educational psychology are arriving at similar conclusions. What are your thoughts about that?
Yes, I think it's a long spectrum with molecular neuroscience, what genes are expressed, and then cellular, and then circuit, and then network neuroscience, and then cognitive neuroscience. And over here, we've got education and educational psychology and psychology and cognitive neuroscience or cognitive psychology. And then, somewhere they meet. And maybe mind-brain education is where they meet, but yes, I think they all are converging. The psychologists prefer to talk about behaviors. And as a neuroscience, I prefer to talk about mechanisms. So the mechanisms, to me, in the brain of what's going on in these cells and synapses and circuits informs how I look at the behavior, which is sometimes very different than the psychologists. The psychologists slice and dice the behavior. You've got episodic learning and semantic learning and declarative learning and procedural learning. Those all happen in those same long loop pathways, and all the long loop pathways go through the striatum and have value calculations. I don't know if you noticed, I didn't emphasize, but midline in my diagram was limbic, meaning emotional pieces, then going through cognitive and sensory and motor. So all of these different kinds of learning that psychologists slice and dice, to me, happen the same way. The mechanisms inside the brain are the same way, they're just different domains. And I think that will happen the same depending on your cognitive domain, whether it's reading or math or history or science. they're all going to have long loop pathways through the value calculating areas of the brain. And some will be more valuable to other people, to some people and not to others. We're individuals. That's great. We can share.
Anne had a follow-up question that I love. Well, I'm not passing judgment, but I think I just did. Sorry. Why do you think students prefer direct instruction when it's less effective than active learning? And she has a follow-up question to that. So I'll ask it, but if I need to re-ask it. Have we conditioned them through traditional transmission models of schooling, or are we reinforcing preference for direct instruction with surface level assessments of learning that students excel at? It's a really great question.
I think her second question answers her first, and I agree with her. Kindergartners, when they're first taught to sit down, are primed to listen to the teacher tell them something. So they learn very quickly, oh, my job is to sit and do nothing and listen. And we've hammered the curiosity out of them throughout everything. So by the time they get to high school, they just want to know what the answers are because they know there's high stakes tests. And then once they get through those, they can, go out and work on putting the prom together or doing the sports event or whatever else they're doing to actually learn how to interact in the real world. They don't care what the exam questions and answers really are, unless somehow or another along the way they've become internally curious or internally rewarded for, I'm good at math. Okay, so I like doing math because somehow or another I'm good at it. Well, everybody can learn math with enough repetitions and enough positive, encouraging feedback. We train reading to criteria. People learn to read and you walk down the street and every sign you read because you can't not read it. We've trained you to criterion, but I don't think we train anything else to that kind of criterion. I think when I was a kid, we might have trained math to criterion, but I don't think we do that anymore. I don't think people know their times tables as well. So it's the system which makes it very hard to get kids engaged because they've been taught that the way you do school is you do well on exams. And other kinds of performances can show you as a teacher what a student has mastered and where they are weak. But the evaluation process is different. And that's where rubrics and other kinds of grading schemes become much more important.
In that example you gave with the kindergartners learning on day one to sit down, they're primed to hear what their teacher tells them. Is there part of the reward circuit is being fulfilled in that when they do it and they get this feedback from the teacher, and that's the same kind of looping that's happening as we engage throughout their secondary schooling as well?
Oh, absolutely, because a social interaction is rewarding. So I got it right. That's rewarding. The teacher told me and I know how to do it. So there is learning in direct instruction. The difference was not that big a percentage. So people do learn, and that's why we argue which is better, because the difference is a small amount. The difference is whether you build the motivation in the student to value their own learning and to want to learn more. I can listen to something and spit it back. As an academic, I've been really trained to do that. I never left the academic system. Most of our students leave the academic system and go out into the real world where they learn on the job training for whatever it is that they're going to do in life. And maybe we've also taught them to be curious about other pieces of our social system other than whatever their profession is. Maybe we haven't, but many of them, I bet, will not go and read physics.
We are getting several questions about published research that you think could be beneficial. People are referencing your 2012 study, and if there's any other studies that you might recommend. So I'm trying to think of how we can give them that information.
All right, so my work with neuroscience and education has all been looking at how teachers utilize this information. And you can go to Google Scholar and Google me, and you can find that. The brainu.org webpage has the very back somewhere, or down at the bottom, you can click to a page which lists my publications as well. I believe all of them are in the public domain and downloadable in one way or another. If you can't, email me and I'll send it to you. I don't think my publications tell you anything more than what I've just said, because I'm not trying to tell you how to teach. I'm trying to give you the rationale for making your own decisions for what kind of pedagogies to use. We can't have students figure out physics on their own through exploration. They need guidance. And the same is true with math and literature and every domain. We give them some context and we ask them to work in that context. So there's a big debate whether you should give instruction first and then set them to doing problems, or doing problems first and then give them some instruction. Well, I personally favor give them the problem first. Give them something to struggle with so that they come up with questions, then do the didactic, and then go back to that problem and give people time to chew on it again, and then bring the class together to share that information, so you get multiple takes on how your content helps to solve that particular problem, and make that problem something relevant to their lives so that they have a stake in it. And especially high school kids are old enough to have stakes, but I know one kindergarten teacher, a pre-kindergarten teacher, who told me, I never thought 3-year-olds could be reflective, but they really are, because she figured out how to give them that agency to reflect on what they were doing, whatever the task was. And, it worked for her to think about how to get the students in a position where they might learn a little bit better and to learn to love learning. You know your audiences. I know, and what I see at the university, which is a different set of students. But I think the principles and the mechanisms of what goes on in the brain are the same at all the levels. But you have to understand that the developing brain doesn't have quite as many connections or complex circuits developed as the adult. But they're getting there, and they're using the same mechanisms.
Part of what I'm hearing you say is I'm going to just kind of recap a few things that one, you talked about how teachers told you the instructional practices. So it's not a neuroscientist telling an educator, here's how you should use this. It's the neuroscientist telling the educator, here are the general concepts. And then the educator in their professional realm and knowledge informing, this is how we can use this information. And it seems that the answer is really that it's not didactic. It's understanding, sort of using these concepts to frame our rationale behind why we're engaging in certain things. So direct instruction is not evil. It's how are we leveraging it? And then how are we also including, thinking about cognitive load, the reward and relevancy, intrinsic motivation. You mentioned interleaving and spiraling, and how all of these things come together, that it's not turn and talk is the answer. It's when are we using these social interactions, and how and why, and how is this helping all of the processes in the brain? Is that accurate?
I think that's a beautiful way to put it. Thank you.
So what we will do is we're going to go ahead and wrap up. I'm going to just sort of let you know about our next session. And then for anybody who would like to stay on and chat with Dr. Dubinsky, we still have a little bit of time, or if you have some more questions that you want to ask personally, you're welcome to do that. We will be sending out information for this session and the recording link, so you'll have access to that. I know we have a participant who was dropping previous research that Dr. Dubinsky was involved in the chat. So we'll try to share that with you as well. So again, thank you so much for joining us today. It's been our pleasure. It's extremely informative, and I hope that this gets the conversation and interest going and continuing that mutual dialogue. Thank you to our interpreters and to everyone who joined us today to make this session possible. As I mentioned, we will share the links and any recommendations from today's AMA by email with everyone who registered, as well as contact information. Dr. Dubinsky, is it okay if we share? I just put you on the spot there. Can we share some contact information for you?
Yes, I put my email address in the last slide, but it's Dubin, Dubin 001 at UMN, University of Minnesota.edu. So I'm totally happy to respond to questions.
Thank you so much. Our next session hosted by IMBIS is going to we're going to have Dr. Sandra Van Alderen, who's a cognitive neuroscientist and teacher researcher. Her aim is to provide insight into the learning mechanisms of learning to teachers from all levels, and that's going to happen on May 30th. So look out for more information about that.